Human health and diseases, we talk about the immune system and immunity. Now, there are, as we all know, there are two types of immunities working in our body, in the human bodies, that is innate and acquired. Innate, which is inborn, which we get since the time, or which we have since the time we are born, and the other type is acquired immunity, which is acquired during our lifetime. That means since after our birth, since after the birth of the human, the human starts acquiring that immunity. That means it starts developing inside the body. It starts developing inside the body as and when we encounter germs, as the body encounters germs. So let us know in details about it. Now, acquired immunity can be broadly classified into two types for our easy understanding, that is active and passive. Now, acquired immunity which will develop during our lifetime can be classified into active and passive. Active is something which our body will develop. Okay, it's a little longer process, so it will develop certain particles, certain molecules, certain cells in the body which will fight against germ. Whereas passive type of acquired immunity is one where the body gets ready-made substances, ready-made cells, ready-made protein substances, which otherwise during active immunity we are producing, but in passive immunity we are getting it directly. So that is known as passive immunity. Both are acquired. Now, first, little bit about the passive immunity. That is, passive immunity, it can be natural, it can be artificial. It is natural. Naturally, all organisms, all living organisms, all humans rather, okay, they get this type of immunity through our mother. Through our mother, after the baby is born, the mother feeds the young one, feeds the offspring. During feeding process, the colostrum, the first milk that is produced, it is laden with antibodies. It has huge reserves of antibodies from the mother, which the mother has produced in her body. It will be passed on to the baby through the milk that she is feeding, through the breast milk. So that is a natural type of passive immunity, natural type of acquired immunity. Why we're calling it acquired? Because the mother had acquired it and then she is just passing it to her young one. The young one is getting the ready-made antibodies. He is not, or she is not producing it, but getting it ready-made from the mother. Artificial type is when some antibodies, those protein structures are injected into the body. Artificially, they are injected into the body when we suffer from some problems. Like problem in the sense that mostly when we suffer from some injury. Okay, we suffer from some injury or snake bite, venom has entered the body, then some antibodies will be directly injected into the body to kill the effect of the venom, the poison, or through the cut if some bacteria, some other microbes had entered, to kill them. Directly antibodies are injected into the body because the human may not have sufficient time to develop immunity, which definitely it will do, but it will require a lot of time, and in the meantime the venom may have adverse effects. So ready-made antibodies are provided. These two are passive immunity. So when you try to understand, always remember passive is something which is given ready-made. Ready-made immunity that is provided to the body is known as passive immunity, whereas active immunity is the point where we have to understand more. Active immunity is the one which is produced in our body. It can be natural, it can be artificial. Whatever type it may be, active immunity will develop only when we encounter germs, when we encounter pathogens, when we encounter antigens. Three terms means the same. They are pathogenic infectious agents. So whenever we encounter them, our body has a natural process, has a process whereby it will start developing immunity. If naturally we have encountered the germ, from the environment, our body will start developing the immunity, or artificially also the germ or the pathogen can be pushed into the body. It can be injected into the body. When it is done, even then, our body will start producing an immune response. So active immunity can be natural, can be artificial. Artificial, we all are very familiar with it, that is vaccination, immunization that we take. Okay, that is an artificial type of active immunity that is induced to be produced. Okay, so let us try to learn a little more. Acquired immunity is said to be very specific. Why is it specific? Because the whatever cells, immune cells develop when we encounter germs, that will remain very specific, particular to attack only that type of germ. A second type of germ will never be attacked by the immune cell that was produced for the first. They are very, very specific. Then two types of responses develop. One is primary response. Primary response is the first for the first time when the body starts producing the immune cells. You have encountered the germ and then the body starts responding against the germ. That will be referred to as the primary response. Then secondary response will be when for the second time we encounter the same germ. That will be called as the secondary response when the body is reacting, also called as anamnestic response. Now, primary response is longer duration. It takes a lot of time because the immune cells have to be produced for the first time. Once they are produced, they will remain in the body. And during the secondary response, what will happen? The second time when you are infected, then already ready-made antibodies are there which were produced in the primary contact, during the primary contact. So what will happen? The second time effect will be almost negligible. Usually the germs will be killed before showing any symptoms. Now, lymphocytes are the cells associated with acquired immunity. Two types of lymphocytes mainly. They are T cells and B cells. T lymphocytes and B lymphocytes. Now, T and B lymphocytes, their origin their origin is always bone marrow. They are always originated in the bone marrow, but they mature at some other location. After originating from the bone marrow, the cells will move, some of them, they will move to the thymus, which is a organ located just near our heart. So it will migrate to thymus where it will mature. Whereas B cells, they will mature in the bone marrow itself. They are responsible for producing antibodies, about which we will be talking. So the immunity in which T cells are mainly involved are called as cell-mediated immunity, and the one where B cells are involved are referred to as humoral immune response. Now, again, furthermore here, as already mentioned, lymphocytes originate in bone marrow. A part of it matures in the thymus region. Here we have the thymus. The thymus is a small organ located just behind our heart. In younger children, the thymus is larger in size. It is larger in size because it is in the developing stage. As one matures, as one grows, the thymus reduces in size, because almost all the T cells, B cells, they will be formed in an early stage of our life. So accordingly, we have the T cells which mature in thymus, B cells which mature in the bone marrow. Furthermore, T cells can be categorized into three types are there rather, that is cytotoxic or killer T cells, helper T cells, and memory T cells. So three types of T cells are produced. Now, whenever the body encounters the germ, T cells, the cell-mediated immunity, will be the first to get activated. T cells will recognize the antigen, instantly modify itself to develop into these three types: cytotoxic, helper, and memory. The cytotoxic cells, as the name suggests, killer cells, they will directly kill the pathogen. They will directly kill the antigen. The helper cells will assist in the killing process, as also it will assist the B cells. It will assist the B cells which are also flowing in the lymph system, in the circulatory system. So these helper cells which can recognize the antigen because they have faced the antigen. So they have the signals for it. They will activate the B cells, and the B cells in turn will produce antibody against the same type of pathogen. So they are in turn related. The third type of T cell, that is the memory cell, T memory cell, what will it do? It will retain the memory that such a type of antigen actually exists in nature. It will retain the memory. So when are all these happening? These are happening during the primary response for the first time. So memory cell has retained the signal that such type of antigen exists in nature. Now second time, if that same pathogen enters the body, memory cell, because it can recognize immediately, so accordingly what will happen, it will stimulate the B cells, also the antibodies related to it, and instantly killer cells will start killing it before showing any symptoms. That is faster comparative, the secondary response I'm talking. So in the primary response itself, we get the formation of memory cells usually. So this is about T lymphocytes and B lymphocytes, the mode in which they work. Now, B cells, as I mentioned, they produce a specialized protein which is known as antibodies. Antibodies also called as immunoglobulin. Globulin is protein. It is a proteinaceous part as related to immune system, so called as immunoglobulin. It is the structure of immunoglobulin or antibodies. It is made up of 4 protein chains, out of which 2 are longer and 2 short. The longer ones are called as heavy chains and the shorter ones are called as light chains. Okay, so the longer ones are called as heavy and the shorter ones as light chains. So in short, we can also write as H2L2. We also write as H2L2. So now this the protein chains, okay, it has a Y-shaped structure. The protein chains, they are linked together by sulfide bridges. So sulfur bonds are formed in between which will keep them attached. Now the tip, okay, we can call this as the tip of the fork. This is like a fork, okay. So the tip of the fork, in both the cases, in both the sides, they are unique. They are unique, like as we have learned about enzymes, they are quite similar to the enzyme. That means they are very specific, like the active side of the enzyme. Similarly, this particular location, which we also call as variable region on light chain, variable region means this varies from one antibody to the other. They are very specific, they are very particular. So one antibody will differ from the other antibody. So that is why they are able to bind to specific antibodies, particular unique antigens, sorry. They are able to bind to unique antigens, the antibodies, due to the presence of this unique location, unique structure. So that is the structure of an antibody. Antibodies can again be of different types. In short, we abbreviate antibodies, immunoglobulins, as Ig. They can be IgA, the one that is passed on from mother to the child. IgM, IgE, which is produced during allergic reactions, IgG and IgD. These are different types of immunoglobulins produced in human body against different types of antigens.